ভাস্কর আর দীপাঞ্জন।
হ্যাঁ আমি করেছি।
আচ্ছা ঠিক আছে।
হ্যাঁ শোনা যাচ্ছে আমার কথা?
হ্যাঁ তোমার কথা শোনা যাচ্ছে। দীপাঞ্জন জয়েন করেছে কিনা বুঝতে পারছি না এখনো। দীপাঞ্জনের জয়েন করার কথা। আচ্ছা ওকে। ভাস্কর একটু আনমিউট করতে হবে, সরি মিউট করতে হবে। Good morning, good afternoon, good evening to everyone, wherever you are attending today's webinar from. Today, we, the Department of Zoology, Navadip Vidyasagar College, is going to organize a webinar on single-cell sequencing technologies and their application for studying brain disorders in collaboration with IQAC of Navadip Vidyasagar College. In this auspicious occasion, today we have different dignitaries, my colleagues, teaching and non-teaching staff of the college, my students, research scholars, many academicians, and others. I, Dr. Shuchishmita Chatterjee Shaha, on behalf of Department of Zoology, Navadip Vidyasagar College, welcome all of you to today's webinar. Firstly, I would like to invite our Honorable Principal Sir, Dr. Shapun Kumar Roy.
Thank you, Dr. Shuchishmita Chatterjee or Chatterjee sir. Thank you. At the inception, I actually express my thanks and gratitude to our discussion, a discussion of attraction. Who is that person? Discussion of attraction, Malosri Moitro. I hope all of us, particularly those who have participated in this webinar, will be enriched, will be enlightened, will gather a huge information related to single cell sequencing. I am a student of commerce. I don't know what is single cell sequencing technologies and their application for studying brain disorder. Brain disorder is known to me, but I cannot explain it. But whole things, that is the title of the topic discussion. Single cell sequencing technologies and their application for studying brain disorder. We have the great pleasure that we have got a person, an eminent speaker who will speak on this topic. I think all the audiences will pay their heed, pay their attention to the discussion of Dr. Moitro, where she is attached to McGill University. Am I correct? McGill University, Douglas Research Center, Montreal. Thank you. On behalf of Navadip Vidyasagar College, on behalf of the governing body of Navadip Vidyasagar College, all the teachers, non-teaching staff or teaching assistant, and particularly the president of governing body, Sri Biman Krishna Saha, although he could not attend this webinar because he is very much busy today. He could not attend this webinar. Thank you everyone. And firstly, those actually are playing a pivotal role behind this curtain is the Dr. Dipanjan Ghosh, who is actually the professor of English, Sudipto Modak, Shubhajit Sen, Nimisha Rai, and Shubham Das. They are very much active and they are actually happy. Whenever they hear that a webinar is going to be held, they actually extend their cooperation assistance. So they are our asset, I can say. And the convener, coordinator of this webinar, IQAC coordinator, Dr. Bhaskar Chatterjee. He is a man who can govern everyone, what to do, what not to do. Dr. Bhaskar Chatterjee, congratulations to you. Joint convener, Dr. Madhuban Dattu. She is away from college about 1 year. Madhuban?
Yes, sir. Not 1 year, sir. Only 6 months.
Only 6 months.
Yeah.
Dr. Madhuban Dattu and Dr. Nirmal Das. Nirmal Das always tries to remain a silent spectator. And organizing committee, Dr. Sujismita Chatterjee. Congratulations to you. Chandrima Mojumdar, newly appointed teacher. Oniket Bishwas, newly appointed teacher. I hope this webinar will be a successful one. I can expect that the webinar will be a great success. We will get a great success, and the participants, the audience, will remain to hear what Dr. Moitro is speaking on. Thank you all. Over to Sujismita Chatterjee Saha or Saha Chatterjee.
Okay, sir. Thank you, sir, for your valuable, nice speech. Next, I would like to invite our IQAC coordinator, Dr. Bhaskar Chatterjee, to Bhaskar. You say something about our webinar.
Thank you. Am I audible?
Yes, you are.
You are audible.
First of all, on behalf of IQAC, I would like to thank the Department of Zoology, all the faculty members, for organizing this international webinar on such an interesting topic. In this, I also welcome all the participants who have come forward to join this webinar. I would like to thank our eminent and distinguished speaker, Manushree Moitro, on behalf of IQAC, for giving us precious time and being with us today. And I think today's webinar is going to be really interesting on such a nice topic, which is single cell sequencing technologies and their application for studying brain disorders. I'm really sure that all our participants will surely get enriched, and we are eagerly waiting for a talk. And finally, I would like to thank you all again and wish the grand success of this webinar. Thank you.
Thank you, Bhaskar. Next, I would like to invite our seminar convener, Dr. Dipanjan Ghosh. Dipanjan, please say something about our webinar.
Yes, am I audible?
Yes.
Good evening, all. Actually, there is nothing to say from my part in this particular webinar because as a student from the arts faculty, so we do not have ample knowledge about this, right? And just like what was said, that is principals are said, that it is about brain disorder. We know about brain disorder, but what actually happening there, it is not known to us. I should congratulate everybody who are present here, the organizing committee, the conveners, and obviously you, Madam Manushree Moitro. Ultimately, in such a situation that we are at this very moment, that is when the pandemic is going on and multiple kinds of problems that we are facing, different kinds of words are coming regarding online teaching and offline teaching, the different kinds of disparities that are there and the problems that we do face. But ultimately, there are some positive sides that we are actually facing at this moment because the students of our college, that in Navadip Vidyasagar College, Nadia, and we can be able to hear your words. It is happening from Canada, and that is obviously something positive that we can find here in this particular form. so eagerly waiting for your lecture, and hopefully we will understand. I don't know whether I will be or not. Thank you, madam. Okay, I wish grand success of this particular seminar.
Thank you, Dipanjan, for your nice speech. Now I hand over the microphone to our colleague, Chandrima Mojumdar. Over to Chandrima. Chandrima? Hello?
Yes, ma'am. Am I audible?
Yes.
Okay, thank you, ma'am. Now I would like to request our departmental head, Dr. Madhuban Datta, to say something about this webinar.
Thank you, Chandrima. But first of all, I want to correct one thing. At present, I am not the head of the department. I am in a sabbatical. I am in a leave, as Principal Sir had pointed out. All credit for organizing this seminar goes only to Dr. Shuchishmita Chatterjee Shaha and my other colleagues, Dr. Nirmallo Das, Chandrima and Oniki. Okay, wishing you all a fine evening or fine morning as the situation exists from wherever we are attending this seminar. I would like to start my few preliminary words with the words of Tagore. Okay, so we all know the famous poem by Tagore, where the mind is without fear and the head is held high, where knowledge is free, where the world has not been broken up into fragments by the narrow domestic walls, and where the words come out from the depths of truth. Being a visionary he was, Tagore had already bestowed upon us the world. He has already envisioned this world where our world will not be broken out into different states. So as Dipanjan was I am resonating the words of Dipanjan as he was saying that today we can hear from Malosri speaking from the far corner of Montreal, and we are sitting in the cozy homes of Navadvip or Krishnanagar or anywhere else, and we can hear her talk about the recent advancement of genetical analysis. Okay, so yes, we are in a pandemic. The situation is grim, but let's not forget the silver lining that only for these advanced technologies in today's world, now we are being able to hear all these illuminating lectures from different corners of the world. So once again, I welcome Maloshri from the behalf of my college and my department as a colleague, not as a head of the department, first of the thing. Okay, so as our science is advancing, we are seeing a plethora of new techniques. And one of this new technique is this single cell sequencing technology, which is helping us to understand various complicated disease processes. Okay, so one such very complicated disease process is neurodegenerative disorders. I think the most complex organ of our body is the brain. Millions of neuronal circuits are there. Millions and trillions of cells are there. Every cell has their own characteristics. Every cell has their own genetical sequences. So in simple words, what this single cell gene sequencing technology is doing to us is helping us to identify each and every neuronal cell. Okay, it is helping us to identify the evolutionary relationship between the cell. It is helping us to identify the mutations, the copy numbers, the methylation patterns, the epigenetic patterns. And in this way, we can identify various kind of cells. We can assign their unique properties by this unique technique, that is single cell sequencing. I also don't know details about this technology because this is a pure research topic, and Malosri will enlighten us more about this technique. But whatever we are saying, that in this stressful situation of today, I think the most dangerous disease that we are encountering today is neurodegenerative disorders. Okay, let's forget about the very complicated kind. Just let's say about the anxiety disorders. Every one of us today is going through a phase of very anxious life. Okay, so these anxiety disorders are also caused by these brain cells. And we all know other neuro diseases like Alzheimer's. We know about multiple sclerosis. We know about autism, okay, schizophrenia. All these are neurodegenerative disorders, and it has been identified that multiple kind of neuronal cells gets affected when we are affected by these diseases. So how the cells can be identified? how the cells gets various kind of apoptotic process actually occurs during these neurodegenerative disorders. So Malosri will help us identify and understand the evolutionary relationship of these cells. So with these few words, I would like to welcome Malosri Moitra to give us an illuminating speech. And once again, I will wish all of us, including me, a happy and fear-free year. Let me say this, let this year be fear-free. Okay. So now I am calling upon Chandrima again to continue the process. Chandrima, you please continue the seminar. Chandrima?
Yes, ma'am. Now I request Dr. Shuchishmita Chatterjee to say something.
Okay, Chandrima. Now, one of our guests, Dr. Corina Nagy. Ma'am, are you here? Ma'am? Malosri?
Hello, yes, I'm here.
Okay, ma'am. Good morning, madam.
Good morning.
She is an assistant professor in the Faculty of the Psychiatry and an associate member of the Department of Human Genetics and the IPN at McGill University and principal investigator at the Douglas Hospital Research Center in Montreal. As an early stage investigator, her interests are focused on the molecular neurobiology of mental health. Her current projects include disentangling the role of specific cell types with particular emphasis on sexual dimorphism in the pathophysiology of major depression. She is a co-supervisor of today's speaker, Malosri Moitra. Ma'am, she is just joining us from other part of the world. Please, ma'am, if you would like to say a few words.
Yes, thank you very much for putting together this very important seminar. I think it's going to be very enriching for everyone. This is certainly a field that is growing and is adding a lot of information to the study of the brain, which is still a black box for most of us. So we are using new technologies to disentangle the role of each individual cell in different pathologies. And Malosri is the best equipped to give you a very nice talk about this. So I'm looking forward to hearing her words as well.
Thank you, madam. My supervisor, Professor Dr. Anilabha Koviraj, is also here today with us. I request to, my supervisor, actually, he is my supervisor, Professor Dr. Anilabha Koviraj. He is waiting for the lecture of Malosri Moitra, and after her talk, he can explain something and he can say something for us. Now I like to hand over the microphone to Chandrima. Chandrima, are you here?
Yes, ma'am.
Thank you. And Corina, madam, thank you for your nice presentation.
Thank you, Dr. Madhuban Datta, for the nice introduction, and also thanks to Corina to say some valuable words. Now, I would like to request Dr. Nirmalo Dash to introduce our speaker, Malosri Moitra.
Am I audible?
Yes, you are audible.
Good evening to everyone and good morning to our speaker, Madam Malosri Moitra and Dr. Corina Nagy. Today, we have a very important person with us, Madam Malosri Moitra. We feel lucky to have her in our midst. It is my great pleasure to introduce our speaker, Madam Malosri Moitra. She is a doctoral candidate in Integrated Program in Neuroscience, McGill University, Montreal, Quebec, Canada. She completed her bachelor degree in the year 2016 from Mount Hockley College, South Hadley, United States. She has received a number of international awards from different international organizations. It includes Bruce Award, FRQS Doctoral Training Award, Dr. Holly Kersley Memorial Award, IPN Returning Student Award, HBHL Graduate Student Fellowship, McGill University Entrance Award. She is specialized in fluorescent-assisted nuclei sorting, qPCR, cell culture, immunohistochemistry, and in situ hybridization. She has also a wide range of working experience in computational biology and single-cell sequencing data analysis. She published a good number of research publications in reputed peer-reviewed international journals with high impact factors. Few journals are Nature Protocol, Pulse One, Nature Neuroscience, Current Behavioral Neuroscience Reports, International Journal of Bipolar Disorders, Neuropsychopharmacology. Her salient publications includes extraction of nuclei from archived postmortem tissue for single nucleus sequencing applications, single nucleus transcriptomics of the prefrontal cortex in major depressive disorder implicates oligodendrocyte, precursor cells, and excitatory neuron, sequencing the human brain at single cell resolution. She has also presented many papers in international conferences in postmortem human brain tissue in depression and suicide, generation and analysis of large and complex single nucleus transcriptomic datasets, establishing a protocol for brain cell type specific epigenetic measurements. She is also member of different academic and research institute and organizations. Due to the constraint of time, I am sorry to say that I could not accommodate all her multidimensional academic and research activities in my introductory part. Without any further delay, I would like to request Madam Malosri Moitro to deliver her presentation. Madam, please. Thank you.
Thank you so much to everybody. Thank you for your very kind words. It means a lot to me. And of course, thank you for giving me this opportunity to speak to you all. I hope that it will be an interesting talk for everyone. And I hope that you will have questions for me and we will have a good discussion after this talk. Especially thanks to all of the audience members for being here today and joining us. So I will begin by sharing my screen. Please let me know if you're able to see the screen. Okay, thank you. So as was very nicely mentioned by many of the professors who spoke in the introductory part, the topic of my presentation today is single-cell sequencing technologies and their application for studying brain disorders. So the presentation is organized in two parts. In the first part, I will introduce single-cell sequencing technologies and give you the background about these technologies. These are the different sections in the first part. So first, I will talk about the rationale, so the reasons and the need for this kind of technology. Then for the most part in this section, I will talk about the transcriptome, so single-cell RNA sequencing, so the study of gene expression at the level of single cells. Then I will briefly touch upon single-cell sequencing of the epigenome, which is basically referring to all of the gene regulatory mechanisms within each cell. And finally, I will mention a few of the resources related to single-cell sequencing data sets that are available right now. So the cell theory, at least in Western science, has been the predominant theory in biology since the 1800s, when scientists like Hooke, Schleiden, Schwann, and Virchow introduced this theory. The main point of this theory is that all living organisms are made up of cells, and cells are the basic building blocks of life. So, as humans, we are multicellular organisms, and we have a lot of cells within our bodies, each specialized to do different functions. You might think that since cell theory has been around for almost 200 years, by this point we would know for sure how many cells are there in the human body. But in fact, this topic itself is still an open area of research, and papers have been published on this topic as recently as 2013 and 2016. So in these papers, people have tried to estimate how many cells are actually there in the human body. They have tried to do this by looking at the mass of the average weight or mass of a human being, by looking at the average volume of different cells. They have adjusted for the different density of cells in different organs of the body. And two of these recent estimates came up with a number of around 30 trillion cells. That is a huge number of cells. And to make things more complicated, when you have complex mammalian organs like the lungs or the liver or the kidneys, there is a lot of diversity in the cellular composition. So the different cells inside these organs have different structures and they have different functions. Right now, we have many international projects ongoing, such as the Human Cell Atlas project, which is trying to identify all of the different cell types and the cell states across different tissues and organs within the human body, and to measure their molecular features, such as their gene expression and their gene regulation. For example, there was a recent publication last year from the Human Cell Atlas project, where they profiled the types of cells in 14 different organs, which are shown over here on the left. And this dendrogram over here is actually showing the relationship between 51 of the cell types that were identified in these 14 organs, and the branches of the dendrogram are showing the relationships between these cell types, and they are colored according to the organ from which these cell types originate. So this is just to establish the fact that there is a huge number of cells within the human body, and that there is also a lot of diversity in these cells, which are very specialized in their form and function. In addition, what adds another layer of complexity is that within the cell, there are a lot of complex machineries present as well. So if we zoom into the cell, we find that the DNA in the nucleus is wrapped in this compact structure called the chromatin. The chromatin itself has a 3D shape or conformation, which affects the expression of different genes that are encoded in the DNA. In addition, the DNA is wrapped around proteins called histones. These histones and this DNA wrapped around a histone forms a nucleosome. The histones in the nucleosome can be modified chemically with post-translational modifications, which can change their interaction with the DNA, and this in turn can affect the 3D structure of the chromatin and gene expression. And finally, the actual bases in the DNA sequence can also be chemically modified in processes such as DNA methylation, which again can affect the binding of different proteins to the DNA and eventually affect gene expression. So there are multiple layers of regulation within each and every individual cell, and multiple complex patterns of gene expression that are produced as a result. So a lot of the information that we have in molecular biology up till now is based on the study of bulk tissues or bulk tissue sequencing experiments. But given that there is such a diversity of cells, what do we realize? One very often used analogy is the smoothie analogy. If we have a smoothie in which there are different types of fruits present, when we study this smoothie, we lose a lot of information about the individual fruits that went into creating this. But if we were to look at the individual pieces of fruit, we would gain back a lot of information about their individual properties. Similarly, the idea is that single-cell sequencing can help us gain back the information that is lost in bulk tissue experiments. We can learn about the cell type specific effects that are masked or lost when we study bulk tissue. And we can also learn about cell type composition changes. So about the numbers and proportions of different cell types in different disease states or under different conditions of biological perturbation. So the main focus here is single-cell transcriptomics, which is the study of gene expression at the single-cell level. James Eberwein is one of the pioneers of single-cell transcriptomics. In fact, one of the earliest papers from his group was published in 2002, where they looked at gene expression in individual human neurons. And this was even before the era of high-throughput sequencing. So they actually used microarray technology to profile these gene expression patterns. Single cells in the field of single-cell sequencing, single-cell RNA sequencing for profiling gene expression was one of the first technologies that developed very rapidly. In the figure on the right, the main steps in this process are highlighted. First, individual cells are isolated, and then usually using oligo-DT primers, mRNA molecules with poly-A tails are captured. The mRNA is converted to cDNA, the cDNA is amplified by PCR, and the cDNA libraries produced are sequenced. One important factor is that unique DNA sequence barcodes are added to the molecules from each cell. This way, in downstream bioinformatic processing, the unique barcodes attached to each cell can then be used to identify which gene molecules are coming from which cells. And so we are able to preserve the identity of the cells and group them based on the type of cell or the cell category that they belong to. Because this technology has been around for almost 10 years now, a variety of methods have been developed in the wet lab for enabling single-cell transcriptomics. Some of the earlier methods involved isolating the cells, perhaps by flow sorting or by micropipetting, into individual tubes or the wells of a 96-well or a 384-well plate. Later technologies also incorporated microfluidic chips in which the cells were isolated in specially designed microscopic chambers. More recent technologies involve the isolation of cells within droplets in microfluidic devices or within very small wells in microfluidic devices. And in fact, the main focus for today is going to be these droplet-based technologies in which the cells are each cell is encapsulated in an individual droplet, and often there is a uniquely barcoded bead that is encapsulated with the cell, such that the barcode on the bead is attached to all of the RNA molecules in the cell, enabling identification later on in the process. And this graph over here is just to illustrate how our capacity for sequencing more and more cells has increased over time. And at this point, we are able to sequence millions of cells using these droplet-based or well-based technologies. In addition to the wet lab techniques that have developed, the analysis techniques have also evolved rapidly over the past decade. Some of the techniques that are used are to reduce the dimensionality of the cell by gene matrices, so to simplify the data in some sense, and then by using these simplification methods, the cells are either grouped together based on the similarity of their gene expression to identify cell types, or in some cases they can be ordered along a developmental trajectory, for example, for someone studying embryonic development, and so on. In addition, methods have been developed that can combine together the data produced by different single-cell experiments, and that can compare the cell types identified in different single-cell experiments. And finally, modern methods are also trying to understand the communication between different cells that are profiled with single-cell sequencing, depending on their expression of ligands and receptors. Now, I would like to briefly mention some of the methods that are available for sequencing the epigenome at the single-cell level. Many of these methods combine single-cell epigenetic measurements with single-cell RNA sequencing. Some of the epigenetic or gene regulation measurements that are performed include measurement of DNA methylation, histone modifications, and chromatin accessibility. Here are some examples of such methods. So there are some methods in which the RNA molecules are pulled down with beads that have oligo-DT primers on them, so that the DNA from the cells and the RNA from the cells is separated, and the DNA can be bisulfite converted and sent for DNA methylation sequencing, whereas the RNA can be profiled for gene expression. Other methods involve the separation of the nucleus for the study of DNA methylation and the cytoplasm for the study of RNA expression. In addition, there are methods that can help profile the chromatin conformation or the regions of open chromatin using enzymes called transposases, which can cut the open regions of chromatin in the DNA. And so, either by isolating the cells in wells and barcoding them, or by capturing them in beads and using transposases along with oligo-DT priming, we are able to find both the RNA information of the cells and the chromatin conformation information of the cells from the transposase binding. There are even methods that have been developed with 3 types of sequencing combined. So bringing together the chromatin conformation profiling or the chromatin accessibility profiling with the DNA methylation and the gene expression as well. So lastly, I wanted to mention some of the resources that are available for all researchers based on these single-cell datasets that have been generated. So one such resource is the Brain Transcriptomics Explorer from the Allen Brain Institute, and this is just a screenshot from their website. And this is showing the expression of a number of different genes in a number of different cell types that they have annotated. And I think this data set consists of around 300,000 cells from the motor cortex of the human brain. Similarly, there is the tabula muris dataset, which is a collection of many thousands of cells from the mouse organs, a variety of organs that are listed over here. So all of these data sets are freely available, and they can be studied by researchers to enhance their own projects as well. So that kind of establishes the background of single-cell sequencing, why we do it, how we do it, and what kind of information we can get from it. The second part of the presentation focuses on single nucleus RNA sequencing of the brain in major depressive disorder. So this is basically the research that I'm working on for my PhD. To give the outline of this section, first, I would like to provide you with some background on major depressive disorder. Then I would like to discuss the methods we used and the results we found when we studied a cohort of male subjects with MDD using the single nucleus RNA sequencing method. And then I would like to briefly mention the new data set that we are working with, which includes additionally a set of female subjects with or without MDD. So first, to give you background, major depressive disorder is a serious psychiatric illness. Some of the symptoms of this disease include persistently low mood, anhedonia, so a lack of pleasure in enjoyable activities, changes in sleep and appetite, and also suicidal ideation, so thoughts of suicide. Depressive disorders affect between 200 to 300 million people worldwide, so that's a huge number of people. In addition, it is one of the leading causes of disability worldwide, especially in people in the younger age groups between 10 to 50 years. So this graph over here actually shows the years lived with disability because of depression among people in different age groups. As you can see, it's highest for the younger age groups compared to the older age groups. In addition, in the medical sense, the treatment for severe or moderate MDD, major depressive disorder, is often antidepressants, but they don't work for all patients and they are very slow to act. So given the huge impact of this disease, it's very important to try to understand more about it. And so, at the molecular level, there are many studies on humans using genetics, transcriptomics, and epigenetics. For example, there was a recent GWAS meta-analysis which identified over 100 specific loci in the genome that are associated with MDD. In terms of transcriptomic and epigenetic studies of the postmortem brain, we have been able to find region-specific and sex-specific changes in gene expression in the brain in depression. Moreover, there have been changes at the molecular level that were specifically associated to early life adversity. So situations in which someone may have faced child abuse and later developed depression. Additionally, since we are especially interested in the cell types involved, there is evidence for an imbalance between excitatory and inhibitory neuronal signaling in the context of depression in the brain. Multiple groups, including several studies from our research group, has shown that there is a change in the expression of astrocytic marker genes and disruption of astrocytic function in depression. Additionally, disruptions in the white matter, so implicating the oligodendrocyte lineage of cells, has also been reported in MDD. So altogether, a variety of different cell types in the brain is expected to contribute to MDD. Finally, there is some level of sexual dimorphism in the pathophysiology of MDD. More women are diagnosed with depression than men. On the other hand, more men are likely to die by suicide compared to women. And there may be social and biological factors that contribute to these differences. There is evidence at the molecular level and at the behavioral level of differences between the sexes, both from human studies and from studies of animal models of depression. So, to elucidate the contribution of different cell types to MDD, we decided to perform single nucleus RNA sequencing in the dorsolateral prefrontal cortex, so one of the frontal regions of the brain involved in higher order cognitive functions, in a cohort of male subjects, 17 of whom were diagnosed with MDD and died by suicide, and 17 of whom were psychiatrically healthy and died of accidents or natural causes. We captured and sequenced almost 80,000 single nuclei. And we actually used this droplet-based approach where these barcoded beads are combined with our cell suspension, and each individual cell is encapsulated in a droplet along with the bead, and the molecules within the cell are tagged with the barcode of that bead for identification later on. These are the main steps that are used for the analysis. First, we cluster the cells into types based on the similarity of their gene expression. Then we annotate the cell types based on the expression of known marker genes. And finally, we perform differential expression analysis. So these are some of our results. This plot is called a T-Sne plot. It is a low-dimensional representation of the gene expression of the cells in our data set. Each dot represents an individual cell, and the different colors represent the different types of cells that we found. With unsupervised graph-based clustering, we were able to find 26 different cell types after quality control. We found more neurons than glial cells, and we found that the highest number of cells were from the excitatory neuronal classes. In this dot plot, the dots represent the expression of different genes in the clusters. So the genes are listed here on the x-axis, and the clusters are listed here on the y-axis. The larger the dot, the higher the proportion of cells expressing that gene, and the darker the color of the dot, the more of that gene is present in the cluster. So we found the expected patterns of mutually exclusive expression of marker genes. For example, genes such as myelin basic protein or MAG and MOG, which are involved in the production of myelin components of the white matter, were specifically enriched in our oligodendrocytic clusters. Similarly, genes such as SNAP25 or RBFOX3 were specifically enriched in our neuronal clusters, whereas inhibitory neuronal markers like GAD1 or GAD2 were enriched in the inhibitory neurons, excitatory neuronal markers such as SATB2 or SLC17A7 were enriched in the excitatory neurons, and so on. So this established that we were able to well identify the cell types of the brain in the postmortem tissue using single nucleus sequencing. In addition, we were also able to identify differential patterns of expression of layer-specific markers in our excitatory neurons. So the human cerebral cortex is organized in different layers, and different types of neurons are enriched in the different layers, and they have distinct patterns of gene expression. So markers like CUX2, RAS-GRF2, PVRL3 were enriched in a subset of our clusters and are likely to represent the upper layer neurons. Markers such as NTNG2, TLE4, NR4A2 were enriched in a different set of our clusters, which are more likely to represent the deep layer excitatory neurons. So once again, our data captured the expected biological patterns in the human cortex. Similarly, for the inhibitory neurons, we were able to identify the expected mutually exclusive subtypes of neurons, such as the parvalbumin positive neurons, the somatostatin positive neurons, and the VIP positive neurons. Finally, we were also able to organize the oligodendrocyte lineage cells in a pseudo-time trajectory. So the oligodendrocyte lineage cells include the oligodendrocyte precursor cells and the mature myelinating oligodendrocytes. Within the brain, it is believed that the oligodendrocyte precursors can gradually give rise to the oligodendrocytes which are mature and myelinating, and thus it should be possible to organize these cells based on their gene expression into a pseudo-time developmental trajectory, and we found that yes, in fact, this was the case for our data set. Over here, I'm showing that across the pseudo-time, the patterns of expression of some genes that are known to be either enriched in mature oligodendrocytes like MAG or MOG or MBP or PLP1 are increasing as the pseudo-time progresses, whereas the expression of genes such as PCD815 or PDGFRA, which are more associated with immature oligodendrocyte precursor cells, were enriched in the early points of the pseudo-time, but not at the later points. Finally, we looked at the genes associated with pseudo-time in the MDD cases and the controls, and we found that among the genes that were specifically enriched in or specifically associated with pseudo-time in only the MDD cases and not the controls, there was an enrichment of apoptotic genes, implicating that there may be an involvement of the oligodendrocyte lineage in depression. We also performed within each of our computationally defined cell type clusters differential expression analysis to see which genes are more highly expressed in the cases or in the controls. So in this plot, the clusters are listed on the x-axis, and over here on the y-axis is the fold change of different genes. The blue circles represent the genes that were significantly differentially expressed between cases and controls. The color of the circle encodes the value of the p-value of the difference. And so we found that there were 16 out of our 26 clusters that contain differentially expressed genes. In particular, an oligodendrocyte precursor cluster and a deep layer excitatory neuronal cluster, excitatory 7, showed the highest numbers of differentially expressed genes. Further, we found that there was not a direct relationship between the size of the cluster and the number of differentially expressed genes, which indicates to us that it is less likely to be a purely technical finding and more likely to be somewhat biological. So we compared our differentially expressed gene lists to the genes that have been associated with depression previously in the literature. For this, we looked at the SciGenet database, and we found that in fact the literature mining, so the text mining of all the published literature in the database, shows that the majority of our genes have a positive association with depression in the database, and in no case was there an indication that the gene wasn't associated with depression in the database. And to be fair, schizophrenia was also the next one on the list, but MDD was still the top hit in the SciGenet database. We also looked at the DysGenet database, which is another database of disease gene associations based on text mining, and we found that 15 of our differentially expressed genes were associated with terms related to depressive disorders in this database. Unsurprisingly, most of the genes that overlapped with depressive disorders from our list were contributed by OPC2 and excitatory 7 once again, because those were the clusters that had the highest numbers of differentially expressed genes. Finally, we looked at some of the predicted protein-protein interactions between the products of these differentially expressed genes. So not just this 15, but all of the 96 that we found. And we found some interesting networks that came up, but one that was specifically interesting was the set of genes that were associated with steroid hormone receptor cycling and with immune function. So this set of genes is especially interesting because the steroid hormone receptor cycling is important in depression, because the steroid hormones are involved in stress response, and depression is known to be associated with a dysregulation of the stress response system. So, based on these findings from the male dataset, we were encouraged to continue to expand this cohort by adding a set of female subjects, especially given that there are known sex differences in the pathophysiology of depression. This seemed to be the next logical step. And so with this combined cohort, we now have a data set of almost 190,000 single nuclei from the dorsolateral prefrontal cortex of the human brain. And again, approximately half the subjects were diagnosed with MDD and they died by suicide, whereas the remaining half of the subjects were psychiatrically healthy. So please ignore this. This is a typo. The remaining half of the subjects were psychiatrically healthy and they did not die by suicide. So they were controls. So this is the process we used for analyzing these data. We reanalyzed all of the data using the same pipeline to make sure that the processing was uniform. We clustered the cells together based on the similarity of the gene expression. We annotated the cell types again using all of these 190,000 cells. And we plan to perform differential expression analysis in each dataset and to compare the results. To show you some of my preliminary results so far, this is a UMAP plot. Again, this is a reduced dimension representation of the gene expression patterns in our cells. And the individual dots represent individual cells, and the colors represent the different cell types. And this is a heat map showing the correspondence between the different cell types we had identified in the male dataset compared to the cell types that we identified in the combined analysis. And as you can see, for the most part, there is a one-to-one correspondence between the cell types we identified previously and the cell types that we are currently able to identify, suggesting that our cell type definitions are stable biological entities and not just because of technical data analysis issues. And this plot is very similar to the one that I showed you previously, where I'm showing that the expression of different cell type marker genes is mutually exclusive and specific, and marking out cell types such as oligodendrocytes expressing PLP1 or inhibitory neurons expressing inhibitory markers, and so on. So as I mentioned, the next steps for this data would be to perform differential expression analysis to compare the cases and controls and to try to find similarities and differences between the male cohort and the female cohort, and to gain more insight into the cell type specific contributions towards major depressive disorder in the brain. So as was mentioned in my very kind introduction, these are some of the relevant publications associated with this work. And with that, I would like to thank a number of people. First of all, I would like to thank my supervisor, Dr. Gustavo Turecki, my co-supervisor, Dr. Corina Nagy, Dr. Matthew Sutherman at the University of Bristol in UK, who has provided me a lot of guidance with the analysis of these data sets, Maria Antonietta Davoli and Jenny Yang at the McGill Group for Suicide Studies for a lot of technical help with generating these data, members of Dr. Yanis Ragoussis' lab at McGill University, again for technical help with generating this data, and Dr. Deborah Mash, who was very kind to provide some of the samples that we have used in this study. And finally, all the members of the McGill Group for Suicide Studies who have been of help in many different ways throughout this project. Additionally, I would like to thank a lot of people at the Navadip Vidyasagar College who have made today's presentation possible. So first of all, Sri Biman Krishna Shaha, President of the governing body, Dr. Shapan Kumar Roy, Principal, who really was very kind in his introduction, Dr. Bhaskar Chatterjee, the IQAC Coordinator, Dr. Madhuvan Datta and Dr. Nirmal Lodash, the joint conveners, and the members of the organizing committee. Thank you very much for making this presentation possible, and thank you to all of the attendees for your kind attention.
Thank you, Madam Malosri Moitro, for your such a nice and valuable presentation. So this is a time for question answering, and all of you who are here with us, please put some questions in both Google Meet and YouTube chat box. And Google Meet session is chaired by Dr. Madhuvan Datta, and YouTube session is chaired by Dr. Shuchishmita Chatterjee Shah.
Thank you, Chandrima. First of all, Malosri, very relevant topic and very interesting, I must say, because suicide and depressions are so much increasing in this stressful society of today. So as I can see that there is no questions, both the questions are from me, and both the questions I am asking from a layman level, because I know the words, maybe I don't know the details. So my first question is, does mental disorder affect the male and female brain tissues differently? Is there any evidence, is there any data evidence that the male and female tissues are affected differently, or are the expressions same? Because you were mentioning something regarding this.
Yes, so in terms of gene expression, it does seem that male and female tissues are affected differently. So when the gene expression changes are measured in different brain regions, in depressed subjects versus healthy subjects, the lists of genes that are differentially expressed in the males compared to the females are not the same. Similarly, in mouse models of depression, the genes that are differentially expressed are not the same. However, people have found that when you look at the pathway level, so not at individual genes, but at the pathways that are dysregulated, there is more overlap. There tends to be more overlap at that level, but not at the level of individual genes.
Okay. And the next one, I want to say that is any specific set of cell more affected? Means you were referring something about oligodendrocytes. Is any specific set of cell more affected during these brain disorders?
So that is one of the questions that we are trying to answer. But in general, there are some overarching theories about how depression develops. One of them is that during development or early in life, stressors can affect how brain development is happening. And so because there are certain areas of white matter in the brain that mature gradually, it is hypothesized that when stressful events are happening in life, the development of these regions is affected. And so things like oligodendrocytes that would be present in these regions, these cell types, would then be affected. But it is more complicated than one cell type because there are also connections between inhibitory and excitatory neurons that mature as the brain develops. And there's evidence indicating that those processes can also be affected by stress and adversity early in life. And so, as you said, it is an open question still, whether one cell type is the most important or whether there's a few cell types or whether they're all equally important is still being determined.
Okay. We have got another question from Horiram Chakraborty. Can ordinary depression become MDD slowly if it persists? And if it is, the answer is yes, how it is happening?
So I'm assuming that ordinary depression in this case means low mood. So being sad or being not in a high and in a good mood. But in this case, the definition of MDD is very specific and it is very clinical. The low mood has to persist for a certain period of time. There has to be a combination of different symptoms for something to be called MDD. And additionally, there are many questionnaires and surveys and scales that are used to assess whether a person will be diagnosed as having MDD or not. So I would say that the ordinary depression is not necessarily in the category of a clinical diagnosis, whereas the MDD is. And ordinary depression, like ordinary sadness, can happen to anyone. That's a normal emotion that humans experience, but only in a few cases where there is susceptibility, where there are environmental factors contributing, people will go on to develop an actual clinical phenotype that will be diagnosed as MDD.
Okay, so you are saying that to be called MDD, there are certain clinical markers. Only then that brain disorder will be MDD. Okay, thank you. Very lucid explanation. At least I understood everything you said. Okay, now over to Shuchishmita for conducting the YouTube question answer sessions.
Thank you, Chandrima. Thank you, Madhuvan and Datta. I have gone through the YouTube chat box, but I have not found any questions there. Thank you, Malusri, for your nice presentation. Now I would like to hand over the microphone to our HOD Madam, Dr. Madhuvan Datta. Madhuvan.
Okay, thank you, Shuchishmita. So the question answer session is over. Now I would like to call our supervisor, Dr. Onilabha Kobiraj. I am saying our supervisor because our respected sir is supervisor of both Dr. Shuchishmita Chatterjee Saha and me, to give us some illuminating words on today's lecture and the topic of presentation. Sir, over to you.
Thank you, Madhuvan. Thank you, Malasi. Very nice talk. And it is nice to see that you are growing in McGill. McGill is a gateway to heaven for academician and gateway to Niagara for non-academicians. You are speaking from a city which now have a minus 14 degrees centigrade temperature, and we are listening here where we have plus 14 degrees centigrade temperature. So in this huge difference, definitely there would have been a huge biological effect on human body. As in ecology, we also study this type of variation in temperature on different forms. Anyway, so in my lifetime, I have seen a full evolution of molecular biology technology, starting from the DNA double helical structure discovery in 1953, just 1 year before my birth, to the next generation sequencing technology, in which I observed in last year, two gentlemen, David Kleinman and one of the Indian, Shankar Balasubramanian, now settled in UK, got the Millennium Award. So it is very nice to see you that you are using some of this latest technology for your research. It is worth mentioning that to understand this technology and to use this for your basic research is definitely commendable. And you have, and as a speaker also, you have mentioned at the initial stage of your lecture how these techniques have gone through over the years, and you have also taken up the recent techniques, that is the droplet-based technology for your research. So I think this is one of the latest technology that are going on for this, and for a layman in biology particularly, so we have got some of the basic information from your talk that we have 30 trillion cells in our body. So I would be happy to know how many cells we shed every day after when we sleep, and next day we wake up in the body. So daily, huge amount of cells are going, and huge amount of cells are going, dying also. But out of that, you have taken a cue from specific cells, 51 different types of cells, and each of the cells have their definite gene expression, specific gene expressions, and you have specifically taken for your research of the brain cells, and for these brain cells, which is a very interesting and one of the recent topic of research for this brain. And at the beginning, many people thought that it is a very interesting talk, because it is you are going to discuss about these brain disorders. So actually you have taken a research on the suicide mode, and this is one of the very interesting topic in our country also, because many people are dying and taking the course of suicide because of several reasons. And one of the basic questions that Madhuban asked, whether there is a difference between male and female, and you have already mentioned that the male have a more probability of undertaking this process, and you have explained it at the genetic level why this is being done, why the males are taking these courses. Okay, very nice way of explaining these facts. I think you will be doing more explanation in futures and doing nice publications. And you have already published very good papers, and I think I have all blessings for you to do a good research and make us proudful here in Calcutta and in West Bengal. Thank you very much, Malas.
Thank you, sir. With this, we are coming to the end of our session, today's session. So I would like to call upon Aniket, our young colleague. So he will now deliver the vote of thanks, and he will also conclude the session for today. Aniket, over to you.
Thank you, ma'am. Very good evening, everyone. Myself, Aniket Viswas, an academic counselor, Department of Zoology. It is a great honor and privilege for myself to propose the vote of thanks on this occasion. First and foremost, I would like to thank our patron, Sri Biman Krishna Saha, President of Governing Body, Navadip Vidyasagar College, for giving us the opportunity to organize an international seminar. I am also very much thankful to Dr. Shopon Kumar Roy, Principal of Navadip Vidyasagar College, for his encouragement, guidance, and continuous hearty support to make this event a grand success. We are thankful to Dr. Bhaskar Chatterjee, IQAC coordinator, for helping us to organizing such a wonderful and memorable event. My special thanks to Dr. Dipanjan Ghosh for his active presence and all-round cooperation. On this great occasion, I feel honored to thank Dr. Anilava Koviraj, the renowned scientist and retired professor of University of Pollani, gracing the webinar as a guest of honor. I am also greatly thankful to Dr. Corina Nagy, Assistant Professor, Department of Psychiatry, McGill University, for her presence. I would like to thank Ms. Malashri Moitro for her wonderful presentation in this webinar, and we are wishing her grand success in life. My cordial thanks to the heads, faculties of various departments and also non-teaching staffs for their sincere cooperation. I especially thanks Dr. Dipanjan Ghosh, Srimati Nimisha Roy, Sri Shubham Das, Sri Sudipta Modak, and Sri Shubhajit Sen for extending their sincere effort and technical support to make the webinar successful. I convey my heartfelt thanks to all the faculties of our department. Last but not the least, I owe my thankfulness to the participants and my beloved students for their all-round cooperation to make it eventful. Hence, we conclude the seminar with a vote of thanks. Thank you, everyone.
Just a minute, Aniket. I sent the in chat box the feedback link. Just a minute.
Okay, ma'am. Thank you.
नहीं आना चाहिए हुर्रे हुर्रे
Amit Shah, please mute your mic. Mr. Amit Shah, please mute your mic. Okay, participants, so the feedback link will be sent both in YouTube and chat box here in Google Meet. You please click on the link and feed on the relevant informations given there, and you will be provided your e-certificates. Well, the technical thing is going on. Thanks once again, Malashri, for a wonderful presentation. I personally enjoyed your talk very much because the topic is so much interesting and so much relevant for today's world. The topic of depression and anxiety and how it is related to our brain disorders. Thanks once again. Okay, so the feedback link has been given. Okay, so the feedback link has been given. Anyone who is asking, please you can go through that. So we can now end our seminar here. Shruti, you please stop the recording. We can end our seminar here.
Okay. Feedback link is also available in Telegram group also.